honor and pleasure to be able to come and give a traditional welcome and a prayer to open sacred space on this particular subject, which is kind of dear to my heart because I know a few people that do have MS. And of course, it's pretty heavy stuff that we're dealing with, and a lot of people in their lives deal with it on a constant daily basis, and you know, our hearts go out to them. And so what I'd like to do right now is I'd like you to imagine yourself standing outside. It may be snowing, might be a little bit of breeze, but stand outside and take a nice deep breath throughout your whole body. Just imagine being warm, but feeling the crispness of the air outside. We are starting to come into winter here. We've had a big snowfall, and it is actually beautiful outside, just beautiful. So we take that deep cleansing breath throughout our whole body, nice and deep. Breathe deep into your body and then let it all go out through your mouth. So as we take that beautiful deep breath that gives life to our body, remembering that the day we were born, we took our first breath of life, and we never stopped breathing for 70 or 80 years. 24/7 we breathe. And what do we call that? What do we call each and every one of you? We call you a miracle. To be able to bring in life through a breath. So just imagine yourself standing outside and you're facing the east direction. And we know that every morning the sun comes up in the east direction, whether there's clouds or rain or snow, we know it's up there. And what does it do? It brings a new day, a new beginning, and new possibilities. And this we are grateful for. So as we take that next deep breath all the way down into our feet, we feel our feet sink into the deep, rich soil of Mother Earth. And there we can smell the richness of her soil. We can feel it between our toes. And there she grounds us into today, into this very moment, being present to what will be taking place over the next hour or so. And this we are grateful for. We, the Lheidli Nation, have been on our traditional lands over 9,000 years as supported by lithic evidence. The changes that have taken place over thousands of years supports the enduring strength, courage, and fortitude of our peoples. Our governance through the Bat'lats brought balance and harmony with our brothers and sisters of other First Nations people. Lheidli clans are Frog, Lasilu, Grouse, Utsut, Beaver, Sa, and Bear Sus. Through this system, we know who our family connections are. Through our oral history, the use of legends told of our travels, our hunting and fishing territories, our trading practices with all peoples. May remember and hold steadfast in what Creator's blessed us with, and that is our traditional territories. Our ancestors have always welcomed people to our territories. On behalf of Lheidli elders, our youth, community members, and chief and council, it is absolutely our honor to welcome each and every one of you to our traditional territories. All my relations must see. So in saying that, we have 40 participants, it looks like. So just imagine yourself in a big room. And in this big room, you're standing in a circle. And you're, before COVID, clasping hands to create a connection of energy running through the circle of life. So if we can bow our heads, we together create a sacred space for what will be taking place. So we call on the Creator and the grandfathers and the grandmothers from the East. Thank you for being with us today and supporting the MS Speaking series. Let us remember the courageous people that are fighting the valiant fight, whose spirits are strong and continue to be strong as they deal with the disease of MS on a daily basis. That beautiful spirits continue to shine in the rising sun of the East. Grandfathers and grandmothers of the South, we ask that you give us the support in creating a future without multiple sclerosis. May our prayers be a constant support in finding the cure. Grandfathers and grandmothers from the West, be with us today in sharing our hopes and dreams for families, friends, and anyone who has been touched by MS. We ask you to hold them when they can't stand, cradle them when nothing but tears flow, nurturing their souls with love. Grandfathers and grandmothers of the North, may your knowledge and wisdom help educate and support those who believe a cure can be found. Mother Earth, may we walk on you softly, leaving no footprints. We are nurtured and sustained by what you so freely give us. May that be returned to you. Creator, give us strength and loving support to all that will be participating in the presentation of multiple sclerosis today. May they be blessed as their hearts are as big as the sky, remembering all the beautiful people whose lives are involved with, support, and share their stories about MS. May Creator hear their story in love, light, and healing. Creator, hear our prayer. All my relations must see. Snachalia to you all. Thank you.
Thank you so much, Darlene, for your special welcome and prayer. Now I will hand the microphone to Dr. Kendra Furber with her presentation.
Well, thank you for inviting me to speak today. I'm quite excited to be connected to you all in some way or another. And thank you, Bruce and Darlene, for such a warm welcome. It's been such a privilege to join the Northern Medical Program here at UMBC on the traditional territory of the Clay-Claytonay. My family has just had a wonderful time kind of exploring the area here, and we hope to explore more of Northern BC when circumstances allow. But I do have to say, being a flatlander from Saskatchewan, I'm still very nervous about all these houses built into the sides of hills. So that's something that that has struck me as different here. But I'm going to get this set up. Bruce, you might have to help me, but let's see what I can do. Where do I go?
On the bottom of there's a toolbar that says share screen.
On the bottom
of your Zoom account, your Zoom screen. There should be a toolbar that says share screen.
No.
Actually, you might have to make Kendra the host.
Let's see. Maybe. Okay, there you go. So now you should have it, Kendra.
Security, breakout rooms. Yes.
So you click on that, and then you should be able to click on what screen you have your presentation on.
Okay, so how are we there? Is everyone?
Looks good.
So in my career, I started at the University of Saskatchewan and then went over to the University of Calgary for my PhD. And I did some very basic research in cell biology. And then I had the opportunity to go back to Saskatchewan. And I was very fortunate to get funded by the MS Society of Canada with a postdoctoral fellowship, which just really opened a whole new avenue of experiences and directions that my research could go, kind of bridging the very, very basic questions about how your cells function, all the way to clinical and epidemiology literature. And so spending my time in Western Canada, even though my family has not been touched personally by multiple sclerosis, I've heard many, many, many stories of the impacts that the disease has on people's lives and their families' lives. So this is the latest data collected from the Multiple Sclerosis International Federation. If you're looking for information, I suggest you go here. It's a wonderful website, as well as the MS Society of Canada has a wonderful website, which I'm sure you guys have all been to. But you can see Canada and the United States, as well as some European countries, have the highest prevalence of MS in the world. And particularly in Canada, the Western provinces have over 300 people per 100,000, which is really the highest rates in the world. So I know this topic, and really the research is happening. It is really dear, kind of to our hearts in this country. So I do have just a couple disclaimers. I am trained as a PhD, so in basic research. I hope you find this session informative, but really for medical advice, specifically with respect to treatments, you need to be seeking guidance from the professionals, your neurologist or primary healthcare provider. The other thing I just want to mention is I am going to talk about animals used in research and the models we use. And I know this can be a touchy subject, but we do a rigorous ethics protocol and get approval from our university, and everything is done in compliance with the Canadian Council on Animal Care. So my acknowledgements, I don't work closely with any pharmaceutical companies, but I do just want to acknowledge the funding sources that have supported my research over the years, specifically the Multiple Sclerosis Society of Canada. And just recently at the UMBC, my lab has been funded by the National Science and Engineering Research Council of Canada. So as well as all my wonderful collaborators and mentors in Saskatchewan, I have a great group of trainees, young trainees here that are helping me set up my lab and get the research started. So I did hear Anna's news clip the other day, and she was kind enough to put in a word for my seminar. And I hope you guys aren't disappointed. What I've learned is people living with MS are very informed. They usually know what's going on, but I hope I can maybe get you excited not only what's happening kind of in the clinical world, but what's happening in labs, and how hard we are working to understand what's going on in the brain and develop new strategies for the treatment of MS. So this is just a brief overview. This is from Compson and Coles, so probably one of the most well-known kind of articles describing multiple sclerosis. And you can see kind of the typical clinical course. It's not the same for everybody, but most people start with the relapsing remitting form of MS, where you see these clinical episodes or attacks. And usually there's almost completely recovery in between. And as the disease progresses, you may not see full recovery between these attacks. And then at some point they progress to secondary progressive, where you see a slow decline in function. And then also we all know that there are people that are diagnosed with primary progressive as well, that look like this, have this disease trajectory from the onset. But if you look at what's happening, kind of, if we try and think about what's happening in the nervous system or in the brain, really you can see these inflammatory attacks in the brain. And it's only when they reach a clinical threshold that we see some sort of kind of episode or attack that will send people for treatment or to the hospital. But these inflammatory episodes are happening all the time, and with advances in imaging, especially MRIs, we have a lot of better understanding of this subthreshold disease now. But what I really want to point out is as the disease progresses, there's less and less bouts of inflammation, and there's more degenerative processes taking place in the brain. So we have a decrease in total brain volume, and much of this is due to an increase in axonal loss. So we'll talk about axons, but they're the projections of the nerve cells that carry signals from one region of the brain to the next. So if we kind of go down to the cellular level a little more, there's a whole bunch of things happening that we can't go through here, but this is what your nerve cell would look like. So that neuron, the cell body, and all the signals come into these little branches called dendrites. And then it sends out, it kind of computes all this information it gets from other cells, and it sends out a signal down its axon, this long projection. So these are what will project, you know, from your brain all the way down to your spinal cord, and then all the from your spinal cord out to your limbs to kind of tell them what to do. And the reason why these nerve signals can travel so fast is because of the myelin sheath. So these cells are oligodendrocytes, the cells that I look at, and we'll talk more about them later, but they wrap the nerve projections in this sheath that insulates them, protects them, and helps the signal move faster, kind of skips between these nodes. So what we have is some sort of dysfunction or dysregulation of the immune system, and normally our brain protects what cells can come in and out, but we have this huge influx of peripheral immune cells, B cells, T cells, macrophages, natural killer cells. They come in and they have some sort of autoimmune response where they start attacking our nervous system. And often this is the myelin sheath, but it can also be the axons as well. Axons can get damaged and transected. So we have other cells in the nervous system, such as microglia and astrocytes, that come to the aid of these damaged, the damaged myelin sheath and the damaged nerve cells. And they try and compensate and they try and clean up. Sometimes most of the time they're trying to be protective, but it can kind of be adapted. It can become a maladapted response. So there's a lot of interest in their role in the pathology as well. And kind of the bottom line is, if we don't get repair of this myelin sheath or the axon, we get progressive degeneration, where we lose these connections between our neurons. And that's really where we start seeing deficits, you know, in motor function or sensory function, or even what's becoming more and more appreciated is deficits in cognition. So just kind of the key hallmarks, I guess, is that this massive inflammatory or dysregulation of the immune system. We see demyelination and we see axonal damage. And as the disease progresses, less inflammatory dysfunction, but more and more neurodegeneration. So this is an article that I really like. It's actually from 2000. So something that's really apparent in the literature right now is whether MS is a heterogeneous disease. And really the big debate is whether it's primarily an autoimmune pathology or a neurodegenerative pathology. But this seminal study from Claudia Lucinetti, who's now at the Mayo Clinic, they showed different patterns of lesions. So these are active lesions right as the demyelination is occurring. And there was a couple that they thought were T cells, so that's your immune cells, and antibody mediated, which they characterized as autoimmune. And then they also saw other patterns which they thought were much more suggestive of a primary oligodendrocyte pathology. So that's the myelinating cells. So really, the question is a chicken and egg question, right? So is it something in the peripheral immune system that initiates an autoimmune response, that then those cells migrate into the central nervous system causing damage? Or is there really something in the central nervous system that's triggering an altered immune response, and this then gets amplified in the periphery, and those cells migrate back into the CNS almost as a defense mechanism, but they're really damaging. They're recognizing our own tissue as bad or foreign and damaging our nervous system. Regardless of really which is the primary cause of the disease, we really need options where we can treat both components, which is kind of where I'll talk a little bit more in terms of the disease-modifying therapies. So is everybody okay here? I'll move on. Again, you can just type your questions, and if there's anything pressing, let me know, and then if not, I'll answer or take discussion at the end. So this is your timeline of disease, the approval of disease-modifying therapies in well, sorry, this is a FDA, so a United States timeline, but it's very similar in Canada. And I want to point out a couple things. So multiple sclerosis was first described by Jean-Martin Charcot, a French neurologist, and this was way back in 1868. And as you can see, over 100 years later is when the first disease-modifying therapy was approved. So up until then, they were trying to treat symptoms and side effects of multiple sclerosis, but they had nothing that really changed or slowed the disease course. So really in the first decade, you had the interferon beta and the glatiramer acetate that came on the market. And then in the early 2000s was when we had actually the first monoclonal antibody that was approved, natalizumab. And then we jump again to 2011. And really it's been in the past decade where we had, 7 novel compounds that really have come on the market. So there's just been this huge amount of progress, I would say. I know it can seem slow, and there's no cure, but really in this last decade, there has become so many more options for treating and controlling multiple sclerosis. And then if we look at each one, this is adapted from one of my papers, but the MS Society has lots of wonderful information on their website that you can find if you're interested in. But what I really want to point out is almost all of the disease-modifying therapies are approved for the treatment of relapsing and remitting MS. So there's a few exceptions where they are approved for secondary progressive MS, but that's when there's only evidence of relapses or those inflammatory attacks still in the CNS. And this was just a huge breakthrough a couple years ago. Ocrelizumab was the first approved drug for the treatment of primary progressive MS. But I just want to note this is still with conditions, and typically that is some sort of evidence of the inflammatory, underlying inflammatory pathology. So if you look down this last column, really the important point here is all of these drugs modulate the immune system, or the response of the immune system. We think a few go into the central nervous system and they might be protective, but we really don't know how they affect the cells in the CNS or in the brain. So either modulating the profile of immune cells or preventing those cells from migrating into the brain. So we've really become quite good at or much better, I should say, at controlling inflammation. What we haven't really had any breakthroughs on is either preventing the neurodegenerative component or actually promoting regeneration after the damage has occurred. And so there has been a real push, and the MS Society has put a lot of money into research, as well as MS societies around the world, into research on finding therapies for progressive MS. So either secondary progressive or primary progressive MS. So that's where I'm going to turn and talk about some of the basic research. So I'm not going to talk so much about what's happening clinically, but really the early stages and how we try and kind of develop these drugs. So can't see too many faces, but I hope everybody's with me right now. Maybe a couple smiles. So in the lab, we use animal models. So that might be the whole animal, or that might be the tissue from animals. And so here are some examples. What we can do is take the cells from a rodent brain, so a mouse brain or a rat brain, and we kind of break them apart and put them into a dish. And so each of these blue dots is a different cell, and these we can image or use markers for different molecules we're interested in. And then you can see some of them here. These are the myelinating cells. They start to grow these really nice processes out, and we can kind of look at how that is occurring. This is where, instead of breaking the tissue apart, we just take a thin slice, and we can lay that out in the dish. And this, you can see, this is a white matter track. So the axon projections and the myelin in part of the brain, and you can see really this nice organization is kept. And so when we work in a dish, it gives us really tight control over the environment that the cells are growing in, is one reason we do it. And the other reason, it's very easy to manipulate these cells. So we can change the expression of different molecules. We can treat them with different drugs. So this is really kind of the first step where we can do some rapid screening to find effects either on protecting neurons or the activation of immune cells, or look at the myelinating cells and how they develop and myelinate. And then really we have to look at things in the whole animal or in vivo. So we can the downfall of this is we can have some artifacts, right? Because the cells aren't really in their normal environment. So we always want to look in the brains of animals to make sure what we're seeing in the dish is also what's happening in a disease model. So I'm going to touch on just two of the most common models here. And one is the EAE model, or the experimental autoimmune encephalitis model. And like a lot of good things in science, this actually kind of happened by accident. They were investigating vaccines for rabies, and they injected tissue from the nervous system into a mouse, and it produced this autoimmune response, and the mouse had paralysis. So what we can do is, this is the most common model used for MS. And we've learned so much about immunology and the dysregulation of those different immune cells from this model. So we put in a little protein from the nervous system, and we boost the immune system. So this is in the periphery, we're inducing this in the peripheral immune cells outside the CNS or the brain. But the mouse mounts a response to these myelin proteins usually, and then the immune cells go in and attack the spinal cord, which is very reminiscent of what we think, or how we conceptualize the underlying pathology of MS. And so we can simply just watch their clinical score, where they have limpness and paralysis starting in their tail and moving upwards. And then we can try different treatments and see if they help that clinical score. And we can also look at the tissue at the cellular level. But as the field has kind of moved to look at repair mechanisms, so once that damage has occurred, can we stimulate the nervous system to repair itself? There's a couple neurotoxic models, and the one that I've used mostly is cuprazone. And we can simply put this chemical in the diet. And so when we add it to the diet, we lose all these mature cells that are myelinating. And so we don't really care that doesn't really reflect what's likely happening in MS. What we want to is remove these healthy cells, so then we can watch and see. You can see these young cells move in, and then they have to develop new myelin. So that's what we're trying to ask, is how can we get these cells to develop new myelin when damage occurs?
Kendra,
Yes.
Amy has a question. She's wondering, so when you inject the cells into the animals, do they actually develop MS, or are you just looking at how the cells react and the myelin work in response to those injections?
Here?
Yes.
Okay, so we don't put a cell, we just put a protein. So those immune cells, the immune cells recognize certain proteins. We call them antigens, it doesn't matter. So something that's in a neuron, or something that's on the oligodendrocyte. So once those immune cells recognize that, then they go into the central nervous system, and they kind of attack wherever they see that protein, they attack it because they think it's foreign and they think it's bad. Does that make sense?
So it's like mimicking, the immune system is mimicking the attack of MS on the cells.
Yeah, so there will be people that will debate exactly how well it reflects the underlying pathology, but that's the idea. That's why this model is used, because it best kind of simulates what we think happens in MS, where we create this autoimmune response that starts to damage the central nervous system.
Great.
Does that make sense?
Yes. Me, it does.
And then, yeah, and then so the other models, we're not so,
Yes, that's what you,
so
that's-
focused on maybe the pathology part of it. We're more just focused on causing the damage in a controlled manner, so we then can look at repair.
Great.
So
Great,
these
thank
ones, we don't really know where the lesions are. They can be but kind of variable, but with these toxic models, we can make kind of just focal damage, and then we know where to look. And we can really just focus on that repair portion. Particularly the Cooper zone I use because it doesn't involve the peripheral immune system very much. So it just lets me focus on what's happening in the cells in the central nervous system. So I think back here I put, your experimental results are only as good as your model system. And the model we use really depends on the question. So there's no perfect model. And really we usually want to compile evidence from all different types of models to really strengthen our hypothesis or strengthen our findings, particularly if we want to move them into translational research. And I'll maybe talk a little bit more about the amount of work that actually gets done before these molecules or these targets actually can move into clinical trials. So what I'm working on here at the university is setting up my program, and we're specifically interested, I think I did this as a reminder, of these cells in your brain or in your spinal cord. So if you're not familiar, the human brain here, we call this outer cortex the gray matter, and that's where all these neuronal cell bodies are. And then they send their projections in, and they can go down to the spinal cord. It can go across to the other side of the brain. So this is the corpus callosum, which is the area we actually look in mice as well. And it's also the area where you find lesions, demyelinating lesions in MS, and the ventricles, so those periventricular lesions. So the axons are sent through here, and this really connects one hemisphere of our brain to the other, which we need to coordinate movement, we need to coordinate our thoughts, our sensory input, just where we are in the world. And so the myelin is this fatty membrane sheath, and that actually makes it look white. So you can visually see, even in a mouse brain, this will look darker, and then this will look very, very white where there's all this myelin. And so the cells in your brain, there's many different kinds of neurons. There's also many different kinds of glial cells. So if you're reading into literature or on websites, the microglia are really the resident immune cells. So they have some similarities with peripheral immune cells, such as macrophages, but they're just found in the brain and the spinal cord. Astrocytes have a whole bunch of functions. They kind of determine what's allowed in and out of the brain. They're involved in the blood-brain barrier. They're also involved in the inflammatory response. And then, like I said, if there's damage, they have this adaptive response that forms the glial scar, where the term sclerosis comes from. And so they do that to protect your brain, but really once that scar is formed, it really is a barrier to regeneration, right? So there's always this balance of, we want these cells to do the good things, but really prevent them from all the signaling that might really compound or amplify the pathology that's already there. And then these are my favorite little cells, the oligodendrocytes. And those are the ones that are producing the myelin. So once these mature myelinating cells get damaged and are destroyed, we need new ones to take their place. So this is kind of a complicated slide, but I just wanted to show you it to see that it's never just one molecule. It's always a whole bunch that work together or against each other. And so it's really like figuring out a puzzle. And so in the adult brain, these are the oligodendrocytes, the myelinating cells again. And you have these immature ones that hang around, and they'll move towards the injury, and then they'll proliferate and make more cells. But what has to happen is they have to transition into this state where they start growing their processes and they start myelinating. And we're interested in a molecule called sirtuin-2. It's shown here, but it works together with all these other molecules. And you can think of this as like a program. So just how we type a program into our computer and it whatever tells, you know, our game or our document or whatever what to do. All these molecules together make up a molecular program, and they tell the cell to become an oligodendrocyte. So they tell the cell to start producing these proteins or lipids or whatever's needed to make that myelin. So this is just an example again of what we can do in cell culture. We can stain and look. So all the red here, the pink, is this molecule we're interested in, SIRT2. And all the green staining are these oligodendrocytes. So we can see the SIRT2 here around in the cell body, the center of the cell. And as that cell matures, we can see it's moved into the myelin, or moved out into all these processes and into the myelin. So the question is really what it is doing and what are the other molecules it's interacting with. And then, so the cell culture is a very good model to ask those basic questions in. As part of my postdoc, and a little bit of what I'm carrying on with is looking in animal models. So with the mice, there's lots of genetic models where they can either make a protein expressed or actually take it away. And then we can ask, how does that mouse function without that molecule or that protein? Or so these are the mice. They actually look quite normal. And then this is the protein. So this is just a check, what we would do to check and say, well, the normal mouse has this protein or this molecule, and then our mutant mouse doesn't have it. And when we look in the brains, so this is cut down the center. So all those axons that go across connecting the different hemispheres, this is what they look like in your brain if you're interested. And all these dark circles are the myelin. So we can see without this molecule, when the animals age, we start to lose the myelin. So we really think it's important for the stability of the myelin. We think it's important for the development of these oligodendrocytes. If we look in our animal models of multiple sclerosis here, so this is the one that's that best mimics what happens in MS, where you have that immune response that comes in and attacks the central nervous system. So the initial response is very similar between the two mice, but when we're missing this protein, you can see there's this elevated disease score compared to our normal mice down here. So, for example, I'm missing my scale, but for example, a lot of the normal mice will start to see some paralysis in the front limbs, but then they revert back and they regain some function and might just have a limp tail or weakened hind limbs. But the CERT2 mice really have sustained kind of damage or a clinical score with their hind limb paralysis. And then this is our cuprosome model where we're looking at the repair process. And when we feed this chemical, this is stained, this red color depicts a protein in the myelin basic protein. And you can see when we feed this chemical, we see a loss, which indicates the loss of the mature myelinating cells. When we take the chemical out of the diet, we can see that the control mice or the normal mice, they're pretty good actually at recovering. They have a lot of re-expression of this protein, indicating there's been remyelination. But the CERT2 mice, you can see they're a lot slower to regain that myelin back. So not only can we visualize it in the tissue, but we can also measure it. So if you guys have heard things like the word PCR is how we measure the genes that are expressed. So that's the other advantage of the animal models. It gives us a lot more flexibility in analyzing the tissue and kind of making correlations of say a clinical disease score with what's happening in the tissue. Where we try to do that in humans, you're just very, very limited really on the tissue that you can get. And that's just ethical reasons, right? So these animal models really inform us not only what the underlying pathology might be in the disease, but how we can really either prevent damage or promote repair. I guess another thing I should say that we have looked in tissue from brains from people that were diagnosed with MS before they passed away, and we can see this molecule expressed where we do see some remyelination. So again, this would be really, really early stages, but it's understanding the basics of how those cells function. So we know how to fix things when they go wrong. So I wanted to end. How am I doing for time? Okay?
Yeah, you're good.
Okay. So this, I guess I'll tell you about a molecule in your brain that's kind of moved along down this timeline from really, really basic studies into actually clinical studies, and you've probably heard of it. And it's LINGO-1. So here is a depiction, we'll go slowly. So this is your oligodendrocyte, or that myelinating cell, right? And this is your nerve cell, that axon. And so with the demyelinating lesion, the old oligodendrocytes have died off or been damaged, and the axon may have been damaged. So now new oligodendrocytes have to take its place and rewrap or remyelinate this axon. And so there's, I guess, 3 key players. And this molecule called no-go is on the oligodendrocyte. And this is a no-go receptor, so it recognizes the protein. And it interacts with this LINGO-1, which is actually on the neuron. And so the idea was So just like we have those monoclonal antibodies that target immune cells, the idea was if we could target this LINGO molecule, because when these three interact together, it inhibits or blocks the new myelin from forming. So if we could inhibit this molecule, preventing it from functioning properly, then we would get rid of this barrier to remyelination. And this is a long story. So I did a quick search. This is a search for NOGO-A or LINGO and myelination. So this is it's just a search engine by the National Institutes of Health in the United States that most people use for scientific literature. And you can see over the last 2 decades, there was almost 600 publications on these molecules. And so just about 100 were reviews. So 500 publications investigating different aspects, whether it be how they work in cells or whether it be if we can take advantage of that for developing new therapies. And so here, if I can find my screen, the first report of the NOGO and its receptor was in 2000. And one of the very first reports of the LINGO being involved in this interaction was in 2004. I'm going to move this so I can. And then several years later, they actually looked at the therapeutic value of blocking this molecule in animal models. And this is really quite, you can see all the people that have helped out on this paper, but they didn't just do one model. Like I said, we want evidence from a whole bunch of models. So they looked in cell culture, they looked in the slice culture, they used that autoimmune experimental model, the EAE, and then they also used toxic models like cuperzone and LPC to look at the repair processes in the brain. And so just recently, now, a decade later, was the phase 2 trial for, I'll see if I can say it, opiscinumab. Is that correct? Maybe you guys better correct me on that. So I think the verdict's kind of out on this. I don't know if it's going to move forward. I won't weigh in right now. So you're maybe saying, well, why? But this is really, I guess, to me, in my career, this is one of the first molecules I've seen go from very, very basic discovery all the way through to a clinical trial. And even though a lot of these molecules don't make it into therapeutics or clinical trials, I just think we're right on the verge. Like just like how in the last decade that we saw so many new disease-modifying therapies to control inflammation, I really, really feel like all the work that's being done and all the support from the MS Society and other organizations into funding, I'm really, really hopeful that we're just kind of there on the bubble and we'll have some of these big breakthroughs where we can really start addressing the neurodegenerative aspects of the pathology and of the disease. And so I guess my last little plug would be, many of you may have heard the catchphrase, from bench to bedside. And there's been a lot of investment in recent years. So they used to call this big gap in translation, where we would learn things, but we wouldn't actually follow through so they would have an impact kind of at inpatient care. And there has been a lot more funding going into this translational research. But just to give you an idea, so NSERC, the National Science and Engineering Research Council, funds basic research, which typical grants are probably $30,000 to $50,000 a year. CHR, or the Canadian Institutes of Health Research, have had a big push for more translational focus to the grant writing and to the studies. And they fund things, you know, at $150,000 to $200,000+ a year. So I just, if you didn't, I don't know, if it didn't really grab your attention today, I just really want you to appreciate the amount of work and the amount of people that are sitting at the bench doing these really basic studies in hopes that someday we'll find something that's really going to impact people living with multiple sclerosis and other neurodegenerative disorders. So I would think this is where the generation of our new knowledge and our new ideas really come from. And I really think the MS Society in particular, I will say again, has put a lot of money into looking at repair mechanisms in the central nervous system, both at the basic science level and also the translational level. And I'm just really hopeful that soon we'll start kind of seeing the fruits of our labor. So thank you so much to all of you for joining me today. I hope I've kind of educated you a little more about what happens in those labs behind closed doors, you know, up at UMBC and other universities. And I'm really happy to open the floor to any discussion. If you have comments or questions, I'll hand that back to
Kendra, I'm going to reclaim host, and then I can check and see if there's any hands up or anything.
Okay. I'm gonna stop sharing, or do you want me to keep sharing?
You can keep sharing. Thank you so much for your presentation. It was very interesting, and we do truly appreciate all of the work all the researchers are doing behind the scenes that we don't see. And you know, that's the greatest goal for the MS Society, is to try and get that cure for MS at the end of the day. So is there any other questions? People are asking if the recording will be posted. We're going to try and find a way to post it without access to download it, just out of copyright issues. I will let everyone know afterwards if we can post it and where it is posted. So hopefully we can get that done. So Rob Rutherford has a question. You can just unmute yourself, Rob, if you want, and ask your question.
There we go. Hi there. Thanks for the presentation. That was excellent. Good to hear. I'm going to put you on the spot.
Okay.
Did you see any improvements or breakthroughs in forecasting? And are you saying 2 years, 5 years, 10 years? And I know that's a hard question, but just some of these new therapies. I mean, I'm on Ocrevus and it's fantastic and it's working. But I'm wondering if any of this remyelination is, you know, gaining speed, or the clinical results are showing, you know, showing efficacy.
Yeah, so I live a little bit more in the basic science world, but just I was trying to update myself a little bit here. There are a few therapies that are reaching, I think, stage 3, maybe stage 2 or 3, and those are more, I think there's a high dose vitamin H, biotin, and biotin we know is really important for oligodendrocyte function. There has been some work with vitamin D and its involvement in the immune system, but also it's important for oligodendrocyte function. So there's things like that are in the works. But in terms of I think the lingo therapy, I'm not sure if it's quite as hopeful as it once was. But again, I would leave that to the experts. So I think there are therapies that kind of help. We don't really know how they help, kind of in the long term, but there's no silver or magic bullet right now, right? So I think it's really going to be a few more years until we see really the same efficacy that we're seeing with the immunomodulatory drugs. I think it's going to be a few years before we really see that in terms of repair.
Okay, and that's what I'm looking to hear anyway. So thanks very much for that.
Krista, you have a question? You'll go ahead and unmute, please.
Yes, thanks, Marcia, and thank you, Dr. Furber. I really enjoyed this, and go Cougars. But I have, and I had to step off for a bit because my phone died. So did you touch on any, like, are you doing any genetic research with the trials that you're doing?
No. So in terms of genetics, I'm assuming you're meaning like underlying susceptibility to multiple sclerosis. Is that?
Yes, like
yeah.
I have a grown daughter, so that's why I'm asking.
Yeah. So I don't, in the lab, we're not specifically looking at that. I knew they try and do more correlation studies or with epidemiology and looking at genetic susceptibility. In the lab, we really use it as a tool. So we have this mouse that I mentioned, and it's modified so it doesn't express a certain protein. So we use it as tools as you know, understanding the function of those genes. But I would have to do a lot more work in actually correlating that with studies that have been done in humans. Most of the genes I think they have identified are immune related genes.
Okay, thank you very much.
Ashley, you have a question now?
Hi, Dr. Furber. Thank you so much for the presentation. I had to jump in and out. I had some technical issues. So I'm not sure if this was already touched on, and if so, I apologize. But I'm a certified herbalist, and I really believe in herbs and how their chemical constituents can help in our own bodies, right? So has any chemical components been derived from herbs, so immunomodulating herbs, that can help with the immune system or repair it like that? Or is it just your focus just on like a chemical constituent that's been synthetically produced?
Yeah, so that's a good question. So I didn't really touch on it much today, but there is a lot of research being done. And I think the MS Society has some information on their websites about alternate modalities or therapies other than just the disease modifying therapies. And what I must say is actually in the animal research, it's really interesting because, you know, even exercise, everybody knows exercise is good for you, but we don't really know how that changes your cells in your brain or affects your cells in the brain. So the same with the herbs or the compounds that we can extract from the herbs. There is studies being done in animals. Even like extracts from green tea, I'm trying to think of some off the top of my head. Extracts from berries. So mostly are looking at, they're interested in compounds with antioxidant properties. Because we know really cellular stress and oxidative stress plays a large component really in the later stages. Anytime your cells are stressed, right, or overworked. So I can't speak to any individual studies, but that's definitely a big topic of interest, and we're doing some work with collaborators at the University of Saskatchewan that's funded by SAS Flax, because they're interested in components of flax, and specifically lipid profiles and the flaxseed, if that would have any benefits in terms of diet,
Right.
for either protecting or promoting repair.
Okay, and then just one quick more question. So back when I was in university and college, they were talking about the similarities, Parkinson's and Alzheimer's, and the demonization of the nervous system that way, how they are treating those, or anything like that being done with MS patients as well, like using those drugs for MS patients?
There has been a bit. So yeah, really we're learning that white matter pathologies are really prevalent across different neurodegenerative diseases. So even the pathways we're looking at on how to stimulate remyelination in multiple sclerosis, they might be applicable to other diseases. So although I will say that you know, each disease has their distinct pathologies, one of the big areas of overlap is one with mitochondrial function, if that means anything to you. The other big area of overlap is dysregulation of glutamate signaling. So that's the excitatory neurotransmitter, so that excites your neurons. And if you have too much neuronal activity, it can cause, it's called excitotoxicity, right? So your cells get overloaded and they can't handle it. So that's, there's some compounds, and again, that have been looked at particularly in Alzheimer's, that they have shown some interest in for multiple sclerosis in the animal models. I'm not aware that any of them have actually moved forward into any sort of clinical trials.
Okay, thank you so much.
Kendra, there's another question. And is there any current research on cadavers with people who had MS? And if so, how would somebody go about donating their brain?
Okay. Yes, definitely. So that one paper I showed by Lucinetti, it was done in postmortem tissue. So it really depends on the disease courses, or if people have to have biopsies for other medical reasons, they can also use that tissue. And those have been very, very informative. There's just limited information, you know, because we can't, it's just one time point that we can look at. But really wonderful work. And so I'm not sure here, if you contact the Northern Medical Program, they will be able to direct you to the right person. I'm not sure who is in charge of the program here, but everybody at every university will have a contact person that if you are wanting to donate your body, I guess, to medical research after you pass away, you do the paperwork the same as you would as for an organ donor or anything like that, and they'll keep it on file.
Any other questions before we wrap up? I think that looks like it's everything. So Bruce, if you want to do the closing, that'd be great.
Well, sure. On behalf of everyone, I would really like to thank you, Dr. Furber, for your presentation and putting your time for us. On behalf of the UMSSC team, I'd like to thank our amazing club advisor and the main driving force behind our webinar series, Dr. Rihanna Robinson. Another special thanks to the MS Society staff, Adrienne Boothroyd and Marsha Spanier, for their amazing support in coordinating this event. And lastly, thank you, all of you, for participating in this webinar. Your participation and engagement means a lot to our team. If you have any sort of suggestions or feedback, please feel free to contact us at the email addresses that Marsha is going to share with you in the chat box. This is the end of the webinar. Have a great rest of your day, everyone.
And I would just like to say thank you for the honor of being your first speaker in this seminar series. And I really hope someday I will get to see everybody's faces in person. You guys take care.
I will follow up the presentation with follow-up email with all those emails as well, if anybody has any questions. Thank you so much.
Thank you. Bye.
Thank you. Bye-bye.
Here.
Thank you. Bye.
Thank you, everyone. Goodbye.